Okay, I think we're live. Just give 10 more seconds for people to join. Great. So hi, everyone. Welcome to this contributed talk session on transcriptomics, where we'll have the opportunity to listen to 3 presentations from Krutik Patel Newcastle University and Oscar Lapuente Santana. And before we start, just a couple of housekeeping items again. If you have any questions, please put them in the Q&A tab in AirMeet, and please be sure to put the name of the speaker you want to ask the question so that we can keep track of that afterwards. Because we'll hear all the 3 talks first, and then we'll have a joint Q&A session afterwards. And in the Q&A tab, you can, of course, still also upvote other people's questions. So finally, a recording of the session will be available in the schedule after it's finished. So with that, I will introduce the first speaker, who is Krutik Patel, who will talk about the TimiRGeN R package for longitudinal microRNA-mRNA integration and analysis. And I think here we'll have a pre-recorded video. So if we can start that, please.
Hi, thank you for inviting me to present today. My name is Krutik, and I will be presenting the TimiRGeN R package, a novel tool for longitudinal microRNA-mRNA integration and analysis. I'll start with a bit of a biological preamble for those who are not too familiar with what microRNAs are. These are small non-coding RNAs that target specific mRNAs for negative regulation. They're complex in their function because multiple microRNAs can target a single mRNA, and multiple mRNAs can be targeted by a single microRNA. However, they have great potential in biomedical research. This is because microRNAs have been found to circulate in biofluids, so have potential as non-invasive biomarkers. One of the best ways of researching microRNAs is by the generation of longitudinal microRNA-mRNA expression datasets, and these are becoming more popular. A recent PubMed search showed a rise in this type of datasets being generated. And this is for several reasons. Longitudinal or time series datasets provide transient detail. Complementary microRNA data provides regulatory information, and the ongoing reduction in the cost of high-throughput technologies means that datasets like these may become more popular in the future. So it's a fair question to ask what software is out there that can deal with this type of data. A thorough search from Bioconductor, SourceForge, online sources, and locally installed softwares found no current tool which could integrate and analyze longitudinal microRNA-mRNA datasets. Thus, it became an aim for my PhD to create a novel tool to analyze this type of dataset. Several characteristics are needed for this tool to be of use to the non-coding RNA research community. It should work after differential expression analysis to be universal in its use. It should be compatible with RNA-seq and microarray experiments. For example, when we're doing functional analysis, the microarray probes should be uploadable as the background data to make the analysis more appropriate for that dataset. It should cater to a range of popular organisms. Currently, several mammalian systems work very well with this dataset. I've tested it on humans, mouse, rats, zebrafish, cow, and dog. It analyzes multi-omic data separately or together, so microRNAs and mRNAs can be combined and then analyzed, or analyzed as separate instances. Network generation is, of course, very important, and it contains metrics to measure individual microRNA-mRNA pairs. This level of specificity was missing in most tools that I showed previously. And it must be applicable after any longitudinal-based differential expression. So if a user uses a pairwise differential expression or uses a cubic spline, this tool will still be useful for them. I created TimiRGeN, Time-Incorporated microRNA-mRNA generation of networks, and it's currently TimiRGeN R Bioconductor package. This has helped me in my own research as a hypothesis generation tool. It has helped me to generate gene regulatory networks and has helped me to reduce big data. It's an open-ended analysis tool, so network data can be exported to Cytoscape or PathVisio. It has several time series pair analysis metrics such as cross-correlation, which measures the difference between two time series, or odds ratio, which is a measurement of the likelihood of one time series influencing another. It can also generate regression models. And it uses several target databases that have been recently updated to filter out microRNA-mRNA pairs, target ScanMirror DB and MIRTAR-base. This tool was recently published this year. So now I'll go through a few pipelines to show how this tool can analyze microRNA-mRNA longitudinal data. We'll start with a pairwise differential expression approach that was taken on a kidney injury dataset. Differential expression data enters as data frames and is filtered in a time point-based manner. Then two options are here for users to find a pathway of interest. Pathway enrichment, which uses overrepresentation analysis, looks for which wiki pathways are enriched in which time points. And temporal clustering. This uses mFuzz to show how different pathways change in behavior over time. Either method is valid to select a pathway of interest. Once we have one, we can look for microRNAs that influence or regulate that pathway. MicroRNA-mRNA interactions can be filtered by the databases shown previously and by correlation. A microRNA-mRNA network can be generated, or genes can be analyzed with hierarchical clustering or be checked over the time course. Longitudinal correlation or regression analyses can also be performed. There's a suite of pair analysis tools that are specific for time series data here. And then network information can be exported to PathVisio or Cytoscape for further in silico analysis. Alternatively, if a user wants to perform non-pairwise differential expression analysis, here we use a breast cancer dataset as an example. Non-pairwise differential expression data can be analyzed using a cubic splines or the LRT method, or DSEC2 or the MARSIC Pro package. All of these tools can generate a confidence feature for each gene, so adjusted p-value or FDRs. It's up to the user to filter out significant genes from their average count or expression data, and this has to be done before putting the data into TimiRGeN. Average expression or count data can be analyzed with overrepresentation analysis to see which which pathways are enriched for the whole time course. All clusters can be made, and pathways can be enriched for these clusters. Again, either method is valid for selecting a pathway of interest. The overall aim of the TimiRGeN package was hypothesis generation from large complex datasets. A normal pipeline may start with data generation, then pre-processing, and generic steps before differential expression, ultimately leading to hypothesis generation, a literature search, and further experiments. With the TimiRGeN R package, we can find a signaling pathway of interest, find microRNAs that regulate this pathway, generate a gene regulatory network for a more focused hypothesis, a more refined literature search, and greater confidence in following experiments. Thank you so much for listening. I'd like to thank the organizers for inviting me to talk, the Shanley and Young groups at Newcastle University, and the Dunhill Medical Research Group for funding my PhD. Thank you very much.
Thanks a lot. That was great. You see all the reactions as well. So as I said before, please put any questions in the Q&A tab, and we'll get to those after all the 3 talks. So our next speaker is Michael Suchek, who will talk about ScanMIR, a versatile toolkit for scanning microRNA binding sites and predicting microRNA-mediated transcript repression.
Yes.
Take it away.
Thank you. Let me just share my screen. So hi everyone again. My name is Michael. I'm a PhD student in Gerhard Schratz's lab in Zurich, and I'd like to now present you our microRNA target prediction tool ScanMIR that we developed together with Fridolin Groß and Pierre-Luc Germain. Now, if you've already heard seconds ago, microRNAs are small non-coding RNAs that post-transcriptionally regulate large part of the transcriptome. They generally bind to the 3' UTRs of mRNAs via their seed region, and thereby induce silencing of these mRNAs. So there's many different types of seeds, and sometimes this pairing of the microRNA can even be supplemented by a 3' pairing. So there's in general very different ways a microRNA can interact with these targets. However, what makes microRNA repression efficiency prediction very difficult is that microRNA binding kinetics do not really resemble conventional RNA-RNA hybridization. Meaning that lots of the current microRNA target prediction tools, as for example ScanMIR, they basically rely on correlative approaches, meaning that they try to infer from the sequence surrounding of the binding site information to predict the microRNA targeting efficiency. Now this all changed then by the end of 2019 when the group of David Bartel published a new paper in which they actually could measure the affinity of purified AGO microRNA complexes to sequences and to 12-mer sequences, and thereby creating direct measurements of microRNA affinities. These they could then further use to construct a biochemical model to predict microRNA repression values, and even went further on to develop a convolutional neural network to also get these microRNA affinity measurements for microRNAs that were not measured experimentally. However, this data has 2 caveats, kind of, why it's not really easy to access and also especially scale. Now, the first of these caveats is basically that per microRNA, we have really a lot of measurements, like over 250,000 sequences and the corresponding affinity values. And second, also the scripts that McGeary et al. Use in their publication are not really written that they are easily accessible to the broader public, nor are they easy to scale. So to overcome this first issue, what we tried is if we could store this information basically not as single integers, but as a linear model, making thereby use of information from the seed region and these flanking denucleotides here. And this works quite nicely, as you can see for the microRNA 122, where we see here the reconstituted affinity values correlate really nicely with the original affinity values. And also for all human microRNAs, we see that overall they have a really nice Pearson correlation from these models to the original measurements. So we can efficiently store microRNA affinities in linear models that we term KD models. And yeah, this led us then basically to program our ScanMirror suite. It's basically a companion of 3 packages. We have the main ScanMirror package that basically allows you to build these KD models from the 12-mer dissociation rates from the convolutional neural network of McGeary et al. You can then use these linear models or these KD models to scan transcripts or sequences and with our find seed match function. And then afterwards aggregate those matches via the biochemical model. We also provide the ScanMirror data package, which basically provides precompiled KD models of all mouse, human, and rat microRNA sequences. And last but not least, we developed a Shiny app basically to make all these functions also available to non-bioinformaticians. And the next step basically we wanted then to do is to benchmark ScanMirror and to see basically how well our KD model-based repression values correlate to actual sequencing data in comparison to the original scripts from McGeary et al. So to do that, we, as I said, we correlated repression scores that were obtained with ScanMirror to RNA sequencing datasets that were likewise obtained by McGeary et al. After microRNA mimic transfection. And what was nice to see that for most of these microRNAs, we're actually in a very similar range as are the original predictions or scripts for McGeary et al. And even further, when we optimize the biochemical model globally, meaning we don't fit it for each sequencing or for each microRNA as they do in the original scripts in McGeary et al., which you can see also our globally optimized model you see in these light green bars. And this still very nicely correlates with the RNA sequencing data, and most importantly, significantly outperforms TargetScan, which you see here in these gray bars, that is currently one of the best target prediction tools that is publicly available. And second, due to the Bioconductor integration, what we can also see that we can scan a lot of sequences with ScanMir much faster than actually with TargetScan or with these scripts from McGeary et al. What we also include in our package is the flexible scanning of the 3' supplementary potential pairing of microRNAs, basically to predict unconventional microRNA binding sites. So when there's extensive pairing of the microRNA towards its target, this can sometimes either lead to a slicing of the target or actually to a degradation of the microRNA that is termed target-directed microRNA degradation, or TDMD. And ScanMir also predicts and basically highlights of length these kind of binding sites. As you can see here for these two experimentally validated examples, the circular CDR1 antisense that gets sliced by microRNA 671, and this long non-coding RNA Serrano that basically upon binding of MIR7 leads to a degradation of this microRNA. And yeah, in the end, I just want to give you a quick overview how you can use the packages. So after loading the libraries, the first thing you can do is to provide a sequence. This you can either do as a DNA string set, or you can use one of our helper functions, get transcript sequence, that is basically built upon the Bioconductor ensemble annotation packages, and makes it very convenient to get the sequence information for any specific ensemble annotated transcript. In a second step, you can provide the microRNA information. This you can either do by specifying the seed region of the microRNA, or by pasting in the whole microRNA sequence, or by loading one of our precompiled models from the ScanMir data package. This information you can then use to basically scan for binding sites, and as a result, you would either get a GRanges object or data frame. And this looks then like this, basically, where you have information on the transcript, the location of the binding site, the type of the binding site, and the affinity value that this binding site has for this microRNA at this point. What we also implemented, especially for large collections of binding sites, is the possibility to store these objects or to convert these objects as indexed FST files. This basically allows a basic handling of objects out of memory, and thereby largely reduces the file size that is held in memory, as you can see here for this example. And it also allows to read in just parts of the object, which is very convenient if you really have a large collection of binding sites. So if this was, for example, indexed as here by sec names, you can just read in basically all binding sites of that particular transcript. And further, with the view target alignment function, you can basically directly align microRNAs and targets at a specific match to see the binding and potential 3' supplementary binding. And last but not least, you can aggregate the matches via the biochemical model to basically get repression information for each transcript. And what I told you in the beginning, ScanMir is also available as a Shiny app, basically to make all these functions easily available for non-bioinformaticians at this homepage here. So yeah, to sum up, ScanMir is now available on Bioconductor nicely as a developmental version. It's, to our knowledge, one of the few microRNA target prediction tools that actually is fully integrated in the Bioconductor framework. It uses KD model-based repression prediction that is currently outperforming other tools such as TargetScan. And also importantly, it allows very easily to scan specific transcripts or including long non-coding RNAs and also circular RNAs for binding site, which is usually not often supported by other tools. And it allows the prediction of unconventional microRNA binding sites, such as slicing sites or TDMD sites. And all our functions are very independent and easily species independent and easily transferable to other species, which makes them really broadly usable. So yeah, with that, I'd like to thank you very much for your attention. I'd like to thank my group, especially Gerhard Schratt, Pierre-Luc Germain, and Fridolin Groß, who helped with whom I together programmed this tool. And yeah, if you would like to have further information, please have a look at our paper on bioarchives. Thank you.
Thank you so much. This was great and excellent time keeping, everyone. And you also see all the great reactions. So again, if you have any questions, please put them in the Q&A tab. We will get to those later. So first, we will listen to our final speaker, which is Oscar Lapuente Santana, who will talk about easier, predicting immune response using quantitative descriptors of the tumor microenvironment.
Can you see my screen?
Yes.
Well, it's a second. Yeah, everything all right? So hello, everybody. My name is Oscar. I'm a PhD student at Eindhoven University in the Netherlands. And today I will be talking about ESIR, which stands for Estimate Systems Immune Response. It's a tool that we have developed to predict anti-tumor immune responses using quantitative descriptors of the tumor microenvironment extracted from bulk RNA-seq data. Understanding anti-tumor immune responses require thinking of tumors as a complex system. The first layer of complexity is the microenvironment surrounding the tumor, and this is so-called the tumor microenvironment, which is basically an ecosystem in which different cell types live together, such as cancer cells, immune cells, cancer-associated fibroblasts, and other non-cellular components of the extracellular matrix. Additionally, the cell states of the state of the cells in the tumor microenvironment are defined by a complex cellular communication between different molecules within the cell itself and among other different cells. And this has been shown to provide tumors with means of resistance to immune responses. Here, we integrate RNA-seq data together with different types of prior knowledge, such as gene signatures of immune cells, gene signatures of signaling pathways, gene signatures of transcription factors, also gene signatures for ligand receptor pairs, and also on the number of cellular receptor and ligands in the cell surface. We do this integration, and we derive, using QuantiSeq, immune cell quantification data. Using Progeny, we derive pathway activity features. Using Dorothea, we derive transcription factor activity features. We also obtain ligand receptor pairs weights and also scores of cell-cell interactions. The main advantages of these descriptors is the lower dimensionality compared to the original transcriptomics data, and also the biological interpretability of their features. We wanted to relate these input features with the response of cancer patients to immunotherapy. However, it's difficult to find datasets with both RNA-seq and response to immunotherapy. So we therefore thought we look for published transcriptomic scores of immune responses. We put together 10 different proxies of immune response here on the table, and we thought of them as different hallmarks of immune response. And we tried to build models that relate these input features with these proxies in a cancer type specific fashion. And we did so using a regularized multi-tag regression algorithm, which basically relates the features. So, for instance, the immune cells features and the different proxies of the immune response that we call here task. And this is a multitask problem because our response variable is given by a matrix instead of a vector. And this is this particular algorithm implies a group elastic net penalty, in which the features are selected. Those features are selected that are common across different tasks. Also, the different weights are assigned differently for this task. We tune the hyperparameters of the model are lambda and alpha, and we tune them using a randomized cross-validation. With this, I mean we split our dataset into training and test set randomly 100 times. So we end up with 100 models. And because this multitask regression is learning a different model for each task, we'll have 1,000 models. And because we are doing this repeatedly for each quantitative descriptor, we'll end up with 5,000 models. This is only for a specific cancer type. And this is basically easier. We built easier using TCGA data of 18 cancer types in a cancer type specific fashion. So we end up with 18 cancer type models. And basically the user will only need RNA-seq data from patients to derive a score of immune response and also to have insights into the biomarkers of immune response. Just to give an example of how this likelihood looks like. So in the figure on the left, we have the easier score for each patient. But this is a data set with 49 melanoma patients from Riaz et al. That were treated with PD-L1 checkpoint blockers. So, and for each patient, we derive 10 different scores that correspond to the 10 different tasks. And the median of these scores is what we call the easier score. And this is an example of the output of easier in the case that we have a data set with non-available response. However, if we know the response to, for instance, PD-L1, we can assess the performance of our model. And we do so on the figure on the right, in which the ensemble curve basically represents the average across the 5,000 models that I mentioned before. We can go further and look into the mechanisms of the immune response. Here I am showing the immune cells features. You can see it on the y-axis. And I'm showing the association of these immune cells with the immune response for the different cancer types. And just a quick example. So you see in the top part, the CD8, representing the CD8 T cells, have a positive association with the immune response. So higher CD8 T cells will have a higher likelihood of response to monotherapy. And on the network on the right, we can also look at associations between our features with certain tasks. We group our 10 tasks in 4 clusters. And we see that the CD8 T cells, for instance, have a higher weight for the cytolytic activity. So we can get this type of insights. Another example of how we can interpret what is going on in our data set. So also here for the real data set, given that we know the response to PD-L1, we can actually look at the biomarkers that are highly represented, have higher values in the responders, in this volcano plot on the right, and also to those that are higher in the non-responders, in this case on the left. Together, to showcase easier, we provide a easier data package, which contains an example dataset with data from Maria Tassan et al., which this processed data was already available via IMIGOR 210 biologist package, but we further process the data and make it available in easier data, which is already available at Experiment Hub. And here we will contain the RNA-seq data. We have TPM values, we have count values, but we also have information on the tumor mutational burden and response to PD-L1. And here, just to showcase what the user will need to use easier. So we'll need RNA-seq data from, in this case, 182 bladder cancer patients that we will fit into easier, but because we have 18 different cancer type models, we also have to tell easier which cancer type the data belongs to, in this case, bladder cancer, to derive the likelihood of immune response and also the biomarkers before mentioned of immune response. Furthermore, given that for some data sets we have the tumor mutational burden available, we thought of how we could exploit this information, and then we based it on the rationale that immune response and tumor foreignness can be complementary hallmarks for a successful immunotherapy. We decide to combine them to get an anti-PD-L1 response score. We combine them using a penalize approach. We penalize those patients with low or high tumor mutational burden. And we also, and another approach could be to perform a weighted average of these both scores. And here on the figure on the right, we can see so in red we have the performance, the area under the curve of the tumor mutational burden. In yellow we have the area under the curve of easier, and we can see how combining these two scores we outperform both giving a better performance. And also, if you check in the middle for the relative weight equal to 0.5, we have the higher performance, which means that complements the idea that both features are complementary. And with this, I would like to acknowledge my collaborators, and especially my supervisor, Federica. And easier is available at GitHub and the development version, and the easier data is also available at GitHub. And thank you all for your attention. I will be happy to take questions.
Thanks so much. This was really nice talks. I'm going to give the audience a little bit of time to put their questions in the Q&A. We have one question for now, so I'm going to start with that, and I'm going to bring it on stage. So this is for Krutik, and the question is whether you can tell us a little bit more about how you generate the networks.
Of course, and I'm happy to. So this is quite a downstream process of the TimiRGeN R package, which I didn't include because of time constraints. But the package has two outputs that can be uploaded onto PathVisio, which is a networking tool. That is a file of microRNAs and a file of dynamic information. With that, you'll have to go on PathVisio yourself and upload those microRNAs and upload the dynamic information, and then you can create a microRNA integrated dynamic signaling pathway, which shows how the genes are changing over the time course of your data and how the microRNAs may be regulating it. And I'll upload onto the chat a few links to show how to do that.
Perfect. Thank you. I hope that's answered the question. So I'm going to ask a question to Michael, and I was interested in the Shiny app, and I was wondering, like, what can the whole analysis be done there, or is it rather like a visualization of the output of ScanMirror, or how does that combine with the ScanMirror package?
It's actually basically the whole package functions also. So you can scan particular transcripts. We now compiled all rat, mouse, and human transcripts for binding sites. You can visualize these directly, and then also aggregate you basically can download aggregated versions where you see the individual transcript repression scores for each transcript and species or microRNA.
Okay. So it's an alternative to running it in the console.
Exactly. It's basically, we provide this because also most of our lab basically don't use Bioconductor R, so just to have it available for everyone.
Yeah, perfect. Thank you. I was also wondering for Oscar, in the very last slide you had, so you combined the easier and the TMB scores with different weights, and you show that, like, depending on the weight, you got different performance. So how would you determine the best weight, the optimal weight?
Yeah, that's a tricky question. So yeah, at the beginning, we didn't know how we could decide this penalty, but just using the different weights, then we just thought looking at the 0.5, which will reflect that both are complement, then we will stick to that one, I would say, which takes both more into account. But we have seen that this is not it doesn't happen always, but yeah.
So it can be different in different data sets.
Yeah, we have seen that.
Okay, so we have a question again for Krutik here. I'm going to put that on. So you mentioned the use of cubic splines for longitudinal analysis. Can you elaborate a little bit if such analysis is done by using functions from another package, I guess, not from TimiRGeN.
Yeah, for example, Limma has a spline-based approach for time series analysis, and I believe you could probably use that for Limma-Voom as well if you have RNA-seq data.
Okay. So that's done externally.
Yeah.
Thank you. So I had another question for Michael, actually. So I was wondering, just out of curiosity, about runtime. Is this something that takes a long time to run, or is it really fast?
It's quite fast now for individual transcripts, and so what takes a bit longer is if you basically run scans for the whole transcriptome for specific microRNAs, but this is also in the minute range still, so it's easy to use. And therefore, so what we supply basically in the Shiny app is this readily run samples or data frames. So if you're just interested in all binding sites or all repression scores for a particular microRNA for a particular transcriptome, the easiest is to go to the Shiny app actually maybe and just download the data from there. But it's also fairly fast, to use in some time if you're interested in particular microRNAs.
Okay. So we have a question for Oscar here. I'll put that on stage. Our lab studies are injury. We know that white blood cells infiltrate the injured tissue between 24 and 72 hours and have RNA-seq from these intervals. So could the easier package provide an informative analysis for this state?
In our lab, we focus on cancer, but immune responses can be also associated with other diseases. But for this special case, I was wondering if you could also use, for instance, we use the tool QuantiSec to obtain different immune cell fractions from RNA-seq data. These are so-called convolution methods, so you could apply those in the 24 hours time and the 72 hours. And if you are more interested, in that case, you will see the different fractions of immune cells in both time points. But if you are more focused on a general immune response, you could also apply ECR.
Okay, that's great. So are there any other questions? No? I think you made everything very clear. So I will thank our speakers again. These were great talks, and thanks a lot to the audience for joining. And I will see you in the next session, or I think now there is a networking break actually. So thanks a lot.